Good morning to all. So yesterday we started, that is the types of eggs. So those are so how many types? Mainly the presence of yolk. The eggs are different types. So those are we are completed in a previous class. The types of eggs and the determinative eggs, indeterminative eggs, and the different types were completed. And today we move on to discuss about the another topic, that is the types of cleavages now. The types of cleavages are based upon the the after the formation of a zygote. So the fusion of the sperm cell and the ova, the resulted to form the zygote. The zygote undergoes a number of the divisions. So those divisions are mainly are considered here as the cleavage patterns. So those are cleavage patterns or so we are going to discuss today. So the types of cleavages. So those are the meridional plane, vertical, equatorial, and the latitudinal. So we are already completed in our previous class, these are four. So one is a meridional plane, vertical plane, equatorial plane, latitudinal plane. So in these meridional plane, the zygote was so deviated from the animal pole to the vegetal pole in the within equal forms of so two halves are equal or formed, and the vertical plane, it was also occurred the vertical division, but the two halves blastomeres or unequal blastomeres are formed in this, and the equatorial plane, so it maybe takes place the horizontally and forms the two equal blastomeres, and the latitudinal also occurred horizontally and forms unequal blastomeres are formed. So these are meridional plane is occurred vertically, vertical plane is also occurred vertically, and the equatorial plane and latitudinal are occur horizontally. The mere variation between these two, that is in meridional plane two equal blastomeres are formed, and vertical plane unequal blastomeres are formed, and equatorial and latitudinal. So these both are occur horizontally, but so the equal blastomeres are only formed inside the equatorial plane, and in the latitudinal plane unequal blastomeres are formed here. That is the main variation between these four. So these are one of the types of cleavages. And now so the cleavage patterns, the types of cleavages are based upon the presence of a yolk. The cleavage is again the presence of yolk, presence of yolk. So again two types. One is holoblastic and meroblastic. The holoblastic cleavage and the meroblastic cleavage. So in these main variation in holoblastic cleavage, holo means so the complete the whole part of the zygote undergoes the cleavage patterns. So why? Because it may not be contain the more amount of the yolk. The yolk is completely absent in the holoblastic cleavage, and the meroblastic cleavage that may contain the more amount of the yolk. That's why. So only some part of the zygote undergoes the cleavage patterns, and the remaining was so it was as usually presented, that may not be going to be divided. So that is a main variation between the holoblastic cleavage and the meroblastic cleavage. By the presence of a yolk, so in holoblastic cleavage may contain the some less amount of the yolk, and the meroblastic cleavage may, so in these the eggs are contain the more amount of the yolk. Based upon these two, the reason these the division was again cleavage, the cleavage is again two types. One is a holoblastic cleavage, another one is a meroblastic cleavage. The holoblastic cleavage is again types. So that was a and the holoblastic is again a equal and unequal. Equal holoblastic and unequal holoblastic cleavages are also there. The equal that was the undergoes the divisions from the equal. The equal holoblastic cleavage is again two types, that is so it was it may forms the equal blastomeres. Equal blastomeres. That's why it was considered as a equal holoblastic cleavage, and the unequal blastomeres are formed. The equal blastomeres, some of the mammals are the example for the equal holoblastic cleavage, and the unequal holoblastic cleavage or examples are amphibians and the fishes. Amphibians and the fishes are the main example for this. And now and again the cleavage was the holoblastic is again based upon the cleavage patterns, based on the reason. So meroblastic we will discuss it later. Why? Because the holoblastic is not completed. After completion of holoblastic we will discuss about the meroblastic cleavage. And next the patterns of so by the absence of the yolk, less amount of the yolk, now the cleavage is based upon the patterns of cleavage. So the patterns of cleavage and that is the cleavage patterns. So that was mainly radial, the cleavage patterns. So that was a radial and spiral, rotational and bilateral. Totally 4 types. So those are radial cleavage, spiral cleavage, rotational cleavage and a bilateral cleavage. So based upon the cleavage patterns, so that was also comes under the holoblastic cleavage. So those are the radial cleavage. So first of all we are going to discuss about the radial cleavage now. So in the radial cleavage, first it is a zygote. Now so it was undergoes the divisions. What was a vertical division to form the 2 cell, and again forms the 4 cell stage, 2 cell, 4 cell, and third division was horizontally occurred. So it is a radial cleavage. So that maybe takes place radially. So that was a So the zygote, it was the first division was vertical, second division was again the vertical, third division was horizontally occurred to convert the cells into the 8 or 16 cell stage was formed. So this type of cleavage was considered here as a radial cleavage. So these radial cleavage was mainly takes place inside the, so that was echinodermata organisms and amphibians are the example for this. Echinodermata and amphibians are example for these radial cleavage. And after completion of this, and next the spiral cleavage. So it is the spiral cleavage. The first division was vertically occurred, and the second division was opposite angle, completely the opposite angle to the first cleavage. The first cleavage was like this manner, and that was a quite opposite manner in these, like in these direction and in this direction. That's why it was completely comes under the radial. So it may have, see this, so that was a completely first so in this direction and a quite opposite direction and in this direction. So in this manner, so first division was vertically occurred and the second division was quite oppositely occurred to the first direction. So that's why, so the cells are converted to, so those are rotated like that was a spiral like movement. That's why this such type of cleavage was considered here as the spiral cleavage. So the spiral cleavage was mainly, and the cells are moved to the, so sometimes completely the cells are moved to the up to sometimes right side, completely right side of the zygote, and sometimes so those are completely moved to the left side of the zygote. So the left side they move, then the condition was considered here as the sinistral condition. If in the zygote the cells are completely moved to the left side of the direction, then what happens is that was considered here as the sinistral like movement that was mainly occurred inside the rotiferans. Rotiferans are the examples here, and the Annelida is also one of the example for this. The rotiferans and the annelidan organisms are the examples. And now the bilateral cleavage. Now the bilateral cleavage. So in this direction, the first was occurred vertically and the second division was occurred directly horizontally. See this. So in this direction what happens is So first division was vertically occurred, second division was horizontally occurred in this manner. So 2 cell stage to the 4 cell stage was occurred here. Then the remaining cells are so moved, and they can always make the mirror image. So the equally, those are separated equally, and one can form the mirror image with the another one. One can make the mirror image with the another. So these are both are the, so see this. So first 4 cells are divided like this. So again 4 cells are again formed at the back side. So then what happens? One can make the mirror image with the another one. Both are completely same and the equal direction, equal manner, and one can make the mirror image with the another. Such type of cleavage was considered here as a bilateral cleavage. So such type of was mainly observed inside the amphibians are the example for this. Amphibians are the main examples. So see this, 4 cells are formed like in this manner, again 4 cells are formed in this manner. So first formed 4 cells, that is a vertical and a horizontal divisions, may give rise to 4 cells. So firstly formed 4 cells can make the mirror image with the another 4 cells form. So that's why 4 cells structure was make the mirror image with the another one. So such type of division was occurred considered here as the bilateral cleavage. So it was observed inside the amphibians. And next the rotational cleavage, that was the fourth one. So see this, it is a 4 cell, 8 cell, and so it was a rotational cleavage. Rotational, the meaning is there, rotational. First division was vertically occurred, and second division was also vertically occurred, and the third time what happens is 2 vertical divisions. After the completion of 2 vertical divisions, then the another next time what happens is the cells are moved to the 90 degrees of angle. So the 90 degrees of angle, say this, the 90 degrees of angle, so those are rotated. So every 90 degrees of angle, so those are always rotated in the 90 degrees of angle. Then what happens is so one of the side of the zygote was completely become empty, and the only another side that may contain the more amount of the cells. So why? Because so if the division was takes place, the cleavage was occurred in this region, then the cells are going to be rotated, moved to the upward direction in the 90 degrees of angle. Then the division was occurred, then after the cells are moved and rotated, and so that's why what happens is only the cells are fused, and so those are completely moved in a one direction. Then what happens is the remaining another side of the zygote will become empty. So such type of cleavage pattern was considered here as a rotational cleavage, rotated, that was moved completely the 90 degrees of angle. It was completely vary from the radial cleavage, spiral cleavage, and the bilateral cleavage. So in bilateral cleavage, so it was can make the mirror image. and so those are moved in the spiral movement and the radially occurred, but the rotational movement only the cells are moved in the 90 degrees of angle, those are rotated. That's why it was considered here as the rotational cleavage. The example, the mammals, some of the mammals are the example for these rotational cleavage. So now the holoblastic cleavage is completed. Now we move on to discuss about the another one, that is about the rotational cleavage, that was the meroblastic cleavage now. So the meroblastic cleavage is again three types: discoidal cleavage, superficial, and again bilateral cleavage. So the bilateral cleavage was occurred inside the holoblastic and also inside the meroblastic. So the main variation between the holoblastic cleavage and the meroblastic cleavage, that is nothing but, in the holoblastic cleavage the entire cell was the entire zygote was going to participate inside the cleavage. Entire part of the zygote undergoes the cleavages. That was completely going to be divided. That's why that was considered as a holoblastic. But here meroblastic, only some part of the zygote undergoes the cleavage patterns. Only some part was participated inside the cleavages. That was only some part of the zygote was only going to be divided, and the remaining part was may not be going to be participated inside the cleavages. That's why that was considered here as the meroblastic cleavage. It was again three types, that is discoidal cleavage, superficial cleavage, and the bilateral cleavage. And the discoidal cleavage, see this, the first one, discoidal. So in these cleavage, see this, so egg, it was a zygote, here the nucleus is there, and see this, see this. So the only, so here is a zygote and nucleus is there, only the nucleus was going to be divided, but not the remaining part. So the whole part may not be participated like the holoblastic cleavage. What happens? The entire cell was going to be divided into the two equal halves, or unequal halves are formed. But here what happens? Only some portion, see this. So here is the egg, only the nucleus is there, where the nucleus is presented, those nucleus was only going to be divided, but not the remaining part of the zygote, but not the remaining part of the zygote may not be participated inside the cleavage patterns. Only where the nucleus is located, that portion was only going to be divided or cleaved. See this, only where the nucleus is presented, such nucleus was only going to be divided due to the presence of high amount of the yolk. So it should be happen, the main reason is a presence of more amount of the yolk, that is the main reason. So that yolk may prevent the cleavages, that yolk may not be going to be support the cleavages. That's why only some part of the zygote undergoes the cleavage pattern, remaining part of the zygote may not be going to be divided. So that is the main variation between the holoblastic and the meroblastic cleavages. So here the first one, the discoidal, means so the nucleus was resembled like a disc shaped, that was presented on the surface of the nucleus. So on the surface of the zygote, that was mainly a disc shaped. The disc shaped nucleus undergoes only the divisions. That's why it was considered as a discoidal cleavage. The main example for this discoidal cleavage, that is nothing but the reptiles and birds. Reptiles and birds are the main example for this. The reptile egg and a bird's egg may contain the more amount of the yolk than the amphibians and the fishes and the mammals. So that's why the discoidal cleavage, it is the discoidal cleavage completely. And now the superficial cleavage. The superficial cleavage. So see this. Say this, the cleavage takes place on the surface of the zygote and the surface part of the zygote region. See this. So, 1 cell to the 2 cells, 4 cells and completely the division was occurred superficially, means on the surface region. That is why such type of cleavage was considered here as a superficial on the surface of the egg and the surface of the zygote. So, such type of cleavage was superficial. The insect eggs are the main example for this. The insect inside the insect zygotes, it should be happen only inside the insect eggs only. That is a superficial cleavage and the bilateral meroblastic cleavage. And the last one, bilateral meroblastic. So, till now we are completed, that is a bilateral cleavage. So, in these also see this, so here also so in these what happens is so totally some part of the zygote undergoes the cleavages in meroblastic and a bilateral between the holoblastic and the meroblastic, that is that was the entire egg zygote participated inside the cleavage, but here only some part is participated only the surface region where the nucleus is presented and the 2 cells or that was a vertical division and the second is a horizontal division. So, 4 cells are formed, that formed 4 cells can make the mirror image. The making the mirror image is the main part inside the bilateral cleavage. That is why it was named as a bilateral cleavage. Here also they can both are make the mirror image, only some part of the egg zygote only participate inside the cleavages. So, it is a bilateral cleavages, it is occurred inside the some of the mammals are the main example for this. And now only last 2 is there, that is a determinative cleavage and indeterminative. So, these are presented in the previous class. Yesterday we discussed those are determinative X and indeterminative X. Here also the determinative cleavage is there and the indeterminative cleavage is there. At the time of occurring of the zygote cell divisions, any cell was going to be damaged, then such type of organ may not be formed on their future embryo. So, that was invertebrates are the example for that. And so, indeterminative means the vertebrates. At the time of occurring of the the cleavage patterns, any cell was going to be damaged, then there was no problem was occurred. So all the organs are as it is formed. Why? Because in the vertebrates more amount of the stem cells are there. The stem cells can able to produce, they can multiply the numerous times, then they can form the, so if one cell is damaged, then so another cells are going to form that particular organ, but that should may not be occurred inside the invertebrates. That is the main example, determinative cleavage and indeterminative cleavages. So these are totally completed. So it is today's topic, that is completely types of cleavages is over. So yesterday we completed, that is about the types of eggs, and now we are completed the types of cleavages. Types of eggs are over, types of cleavages are also over, and the next you are having in your syllabus. So that is mainly the main topic, that is the gastrulation inside the amphibia. So inside the amphibian, the gastrulation in amphibia, and how the in amphibian organisms at the time of gastrula stage of cell division, how the organs are developed. So that is the main topic, and what is also one of the important essay. So we will discuss it tomorrow. Today's topic is over. That was mainly about the cleavage patterns. We will meet tomorrow.